Chapter 1, Section 1, Cellular Foundations. Now, thinking back to the intro lecture, cells, fundamental unit of life, okay? Cells are going to come in different shapes, different sizes, different complexities. You see here on the left. On the left, a bacterial cell, prokaryote. How do I know it's prokaryote? You do not see a nucleus, okay? I don't care what any textbook ever says you've ever read. My definition in any class you take with me. The definition of prokaryote versus eukaryote, does it have a nucleus? No nucleus, it's a prokaryote. Because if you look at this list of other things there, you know, cytoplasm, plasma membrane, ribosomes, some form of genetic material, DNA for pretty much everything that's not a virus. Every living cell on this planet has that. There's a plasma membrane, there's some form of ribosomes, there's some form of nucleic acid material. That's the genome, the blueprint. What separates the bacteria from animal, bacteria from plant, bacteria from fungi? Is there a nucleus? That's it. Because I can find eukaryotic cells that don't have an ER, that don't have a Golgi, that aren't going to have some form of internal membranes, but they will have a nucleus. So that is my definitive separation. Now, this picture, if you look right here, it is a little deceiving because you look at it and the prokaryote, the bacterial cell, is about a third the size of the animal cell until you look at the numbers, the bars, 1 micron, 20 to 50 microns, okay? Keep that in mind. The bacterial cell, the prokaryote, the archaea, for all intents and purposes, and yes, there are exceptions to the rule, and no, we're not going to worry about those exceptions. For all intents and purposes, they're going to be greatly reduced in size compared to eukaryotic cells. Now, some of the things that are unifying for all living cells is this thing of a plasma membrane. Okay, all cells have to have some form of plasma membrane. The plasma membrane is what determines inside and outside of the cell. Excuse me a second here, light's acting funny. There we go. It's the boundary. Inside, outside, outside, inside. Some things get through, other things don't. Think back to cell and molecular biology. Okay? Whole chapter just on membranes. They define the cell. Okay? They are going to be made of phospholipids. They're going to have certain proteins associated, peripheral, inserted through them, integral, or small polypeptides attached to said phospholipids, proteolipids. It's a thin layer. Outside, inside, inside, outside. Some things get through, other things don't. But it is the boundary. Internal, you have cytoplasm. Okay? That is this aqueous environment. Well, I say aqueous, toss up some air quotes there, because it's not true water. It's what is referred to as cytosol. An aqueous base and a water-based solution of different salts, amino acids, sugars, polypeptides, nucleic acid, nucleotides, all sorts of stuff in there. What separates cytoplasm and cytosol? Cytosol is what's part of the cytoplasm. The cytoplasm contains the cytosol, any and all ribosomes, nucleic acids, proteins, polypeptides, organelles if it's eukaryotic, that may be present. Bacteria archaea, it's going to be a singular circular genome, generally measured in the millions of nucleotides. The DNA nucleotides, the A's, the G's, the C's, the T's, that are present there, adenine, guanine, cytosine, thymine, look pretty much the same as the A's, the G's, the C's, the T's that are going to be present in you, your dog, your cat, your goldfish, the tree outside. For prokaryotes and archaea, since there are no internal membranes, there's no nucleus, hence prokaryote. That single circular genome is going to be compacted down, and it's going to be housed in an area within the bacterial cell, the archaea cell, referred to as the nucleoid. It's a region of that cell cytoplasm that's going to be rich in nucleotides and any and all proteins that associate with nucleic acids. For us eukaryotes, protist, fungi, animalia, plantae, whatever, our genome is generally going to be either a single circular, large circular, for our protozoan, ancestral cells, or for us it's going to be in multiple linear chromosomes, all housed within a dual membrane structure we refer to as a nucleus. Prokaryotic genomes measured in the millions. Eukaryotic going to be measured billions. B for us, for plants. Number goes down as you get into the more single cellular protists, but it's still going to be 10 to 1,000-fold larger than what you find in the average bacterial cell. And then you look here, the shape, the size. I mean, we're still talking quite small, microscopic. And students always ask, why do cells have to be so small? Why can't they be bigger? Instead of me being trillions of cells sitting here, why not 1,000-fold larger cells? And I can be just billions or, millions. Why trillions? Well, it all has to do with this second bullet here. Transport. If you have a cell, boom, boom, okay, this cell, whether it's really small or really large, let me get this into frame so you can see what I'm talking about, large. Something that enters the cell, passes through the cell membrane on this side, still has to get over to this side. There's no freight carrying organelles and things that are going to be able to do that for things such as oxygen, individual glucose molecules. Yes, for us eukaryotes with internal membranes, we can have vesicles that can move things around, but those are usually things that are after they've been manufactured and processed by the ER and the Golgi. I'm just talking uptake of oxygen, water, Glucose. Getting rid of waste. If the capillary bed's over here, and you've got this really big cell, things diffuse in, or maybe there's some active transport, getting it across the plasma membrane, and then it has to be diffusion that gets it across. Now, if this cell's huge, that diffusion's going to take time. Any metabolic activities over here on this side of the cell, it's going to take time. Chances are those metabolic activities can run out of substrate. Anything over here, any organelles over here that are oxygen dependent, well, that oxygen, as it's slowly diffusing, can be used up by every other organelle and every other metabolic activity. Any waste products. Turns out waste products, as they build up, can be just as detrimental, if not more so, than low oxygen levels. Well, those waste products have to pass all the way through that large cell. Whereas if you have small cell, another capillary bed, small cell, another capillary, boom, boom, boom. The diffusion across this small cell, instead of measured in minutes to hours, the time frame is now minutes to seconds. And that's what it is for us eukaryotic cells. For the smaller bacterial cells, rudimentary transport mechanisms. A lot of times it's, porins opening and closing, allowing things to move in and out faster is the difference between slow diffusion processes. And by keeping the cells small, diffusion across the cell, the cytoplasm occurs faster, metabolic activities can stay and remain at peak instead of this up and down because of the slow diffusion of larger molecules. Now, you've heard me say bacteria, Archaea, Eukaryote. For those of you who don't quite remember, these are going to be the big three domains. Okay, eukaryotes, anything with a nucleus. Okay, I mean, you see entamoeba, the slime molds, the fungi, the animals, the plants, the single cell protists, things like that we're used to discussing. Bacteria, well, these protists, these prokaryotes, not protists. These prokaryotes, you've heard, staph, E. coli, things like that. A lot of times students will ask me, well, where does where do the Archaea fit between the prokaryotes and the eukaryotes? Well, as you see here in this tree, the Archaea are this in-between. Okay, they're not eukaryotes. They do not have internal membranes, but they have structures within their cell walls. They have structures, they have domains, they have genetic things going on that they're not true prokaryotes, the bacteria. They fall in between. So you notice down there that branching, boom. Prokaryotes to the left, eukaryotes ultimately to the right, but somewhere early on that right-hand branch also we think gave us the Archaea. So genetically they're closer to us than the prokaryotes, to the bacteria, but they're really not like a common ancestor of us. So please do not start thinking that way. Just a couple of billion years ago when things were just starting, there was a branching. When we look at living organisms, we like to pigeonhole, group like with like, because it helps us be able to understand things within the group if we're grouping them like with like. group them like with like. So that way if we could find something new, some new organism, and we know it goes into this pigeonhole, which has these other things around it which are quite similar to each other. So if this fits here, then it's got to be like them. So it helps us make some leaps of logic to help start discussing what that new organism could be like. One of the easiest to do is, do they need oxygen? Yay or nay? If it's yay, they're like us. Aerobes. Yay! Breathe in deeply. Thank your nearest tree. If they don't need oxygen, then they're anaerobic. They do not need oxygen as their final electron acceptor in their, cellular metabolism, their energy production. They use sulfur, or they use some other nitrogen, sulfur-containing compound. So instead of oxygen, hydrogen coming together with that now energy-depleted electron in, at the end of the electron transport chain in the mitochondria, forming water, they use some nitrate, nitrogen-containing compounds. Sulfur makes sulfur, sulfur dioxide or something else. I'm not sorry, not sulfur dioxide, some sulfate-containing compound. And then we see that, aerobes, anaerobes can then be subsectioned. Do they need a lot? Can they get by with just a little? Is oxygen there, doesn't bother them, they can use it or not? Don't care. Facultative. Oxygen is actually quite damaging. Keep it away. Obligate anaerobes. So you have to keep in mind that even though there's quite a lot similarities, quite a few differences, everything has some form of metabolism, okay? This maintaining of imbalances that drive redox reactions. Hot potato. Who's got the electron? Where did it go? Has it been reduced? Has it been oxidized? Gains the electron, it was just reduced. Loses the electron, it was just oxidized. How they get the energy for this to occur, as you see here in this list, phototrophs means they use sunlight. Chemotrophs, like us, they derive energy from chemicals. Heterotrophs, they have to take it in. Autotrophs, they do it themselves. So if you stop down and you break it down and you look at it, we are chemoheterotrophs. We have to take in organic materials, biomolecules, chemicals, which we then break down. The carbon-carbon atom, carbon-carbon bonds to get at the chemical energy there to drive the redox reactions, which will ultimately give us. Eukaryotes have internal membranes, nuclear envelope, Golgi, endoplasmic reticulum, and whatnot. Well, it turns out prokaryotes also have a few things that we eukaryotes don't. They're going to have what is referred to as a cell envelope, cellular envelope. That plasma membrane, and then some things just outside of that. Some form of cell wall to help maintain its overall shape. Because very few bacteria are pleomorphic, meaning they can have different shapes depending upon their environment. The majority of them are going to have some form of genetic instructions to be rods, spheres, something in between, some form of corkscrew, whatnot. Bacilli, cocci, cocobacilli, and whatnot. So that cell wall, which is composed primarily of sugars with some polypeptides, is going to be nice and structurally firm. It's not going to yield to osmotic pressures. Because remember, bacteria can't get up and go get a drink when they're feeling a little depleted, a little dehydrated. Water goes in, water goes out based on whatever environment they're in. They have no control over that. So osmotic pressures, water moving in and out, means that if it wasn't for that cell wall, that bacteria would shrink and expand and burst depending upon which way the water was moving. And you look here. Yeah, the microbiologist means kind of, having fun with this because this is what I live for. Gram-negative, gram-positive cells. What does that mean? Well, it's just the results you would get on a specific type of stain. The stain is going to be blue, positive, red, negative, based on whether or not you see a thick cell wall. If it's a thick cell wall, the blue stain will be retained, gram-positive, staph, streps, things like that. If it's a thin cell wall, as you see there with the gram-negative, it's a thin peptidoglycan layer surrounded by twin membranes, the plasma membrane and outer membrane. Well, it's not going to be able to retain the blue. It's going to stain red, thus the negative, gram-negative. Like you would see with E. coli or, oh my gosh, I can't think of anything else. Wow. I really need to up my caffeine intake if I cannot think of any other gram-negatives other than E. coli right now. We're not going to worry about it. But if you look here at the bottom, Archaea. Archaea, they have a glycoprotein that's not quite peptidoglycan, and they've got a pseudopeptidoglycan that's not quite the peptidoglycan we see with gram-positive, gram-negatives. Like I said, Archaea's kind of pro-eubacteria, but not really. But whether you're talking gram-positive, gram-negative, Archaea, eukaryotes, it's all based on the same set of biomolecules. Let's talk E. coli, all right? Our friend E. coli. Well, if you look inside it, you're going to see ribosomes. Okay, you got to have ribosomes. Ribosomes are what are responsible for translation, the reading of the messenger RNA, the making of the proteins. There's going to be a bunch of polypeptides, enzymes that do the metabolic activities. There's going to be metabolites, things that are, the substrates or the intermediate substrates. They're going to be the product or the intermediate products, which are actually also intermediate substrates for the next set and all sorts of weird things. Cofactors, which aid the enzymes too to get the correct shape so they can have a correct function, all sorts of stuff. What's really cool with prokaryotes, they have accessory blueprints. We call these plasmids. These plasmids are going to be small little circular DNA things. Remember I told you the genome for most bacteria measured in millions? Well, these plasmids are such small circular things, they're measured in the thousands. It's only maybe a handful of genes present, but that handful of genes can be the blueprint on how to make a toxin, how to make some form of enzyme that's going to break down antibiotics. Penicillinase, which just basically cuts one bond in penicillin and renders it just nothingness. Or making of antibiotics themselves. Yeah, turns out most of the antibiotics we use nowadays are actually taken from bacteria. Mother Nature's very own. These things have been around 1 billion years longer than us. They have been fighting for space, fighting for nutrients longer than we eukaryotes have been here. Yeah, they've got the best defenses. They've got the best weapons against each other. And then you look at us eukaryotes, all those organelles that you've learned about since you were a kid, mitochondria, endoplasmic reticulum, which you'll hear me refer to as the ER, smooth ER, rough ER. The Golgi that takes things made in the ER and does the final twists and tweaks to turn them into whatever. Peroxisomes, lysosome, granules, and all sorts of fun things. It's so much fun. Then the plants also have their own little things that are only plant. You can find everything from the last slide there, yes, but then a few extras, vacuoles, chloroplasts, the reason why plants are green, the chlorophyll inside the chloroplast that is responsible for photosynthesis. So you go and you look at a nice, cool picture like this one right here. You see that, for the most part, eukaryotic cells, depending upon whether or not you're talking Animalia, fungi. Here you see plantae. There are going to be a few things that are for them and them only. Yes, if you want to know what else there is, take, like in protists and amoeba and those, yes, take another course with me down in biology. What's interesting is one of the first things we've learned to do when we study cells. I mean, yes, coming up with, the microscopes, better microscopes, stains to allow us to see cells and see the different things in a cell. It was great. That was a great first step. But the advent of better, faster, more reliable centrifuges, boom, whole new worlds opened up. We allowed to do, as you see here, what is referred to as subcellular fractionation. So instead of looking at one cell and trying to discern all the different things going on in that cell because it's inside the plasma membrane still and it's just too much there, let's go and start separating stuff out based on size, based on, whether they're positive or negative buoyant in a certain sugar solution. Oh, yeah. Do things float? Do they sink in this solution? And if they sink, where do they sink? Do they sink all the way down to the bottom under centrifugation, or are they held somewhere in between because we're using multiple densities, multiple molarities of sugar solution that have been layered on top of each other? So it's very thin, thick and viscous down at the bottom. Oh. So we can now go through, and as you see here, based on, what solution we're spinning, what speeds, higher speeds, more gravities, low speeds, as you see here at the top, 1,000 Gs. Higher speeds, look at that, 150,000 Gs. Oh, yeah, you can start separating out a lot of different things in different ways. And look what's being separated out. Whole cells up at the top here. Down at the bottom, you're now getting, individual ribosomes. Ribosome, large subunits, small subunits, macromolecules, individual proteins, things like that. Next one up, you're starting to get fragments of individual organelles. Next one up, you're getting whole organelles, whole ER, the whole Golgi. The Golgi is itself and everything in it. So you can now study just what's happened in the Golgi instead of being stuck looking at the whole cell itself. And what's also interesting is cells have their own cytoskeleton, okay? Think back, 240 downstairs in biology. You saw this, actin filaments, microtubules, microfilaments, the in-between intermediate filaments. The microtubules have their own job versus the microfilaments versus the intermediate filaments. Some push out, some pull back, some are there as a meshwork so that the organelles can be anchored in place. One of the things I remember back when I was an undergraduate in the mid-90s was the fact that they started to come out with these new dyes. These new dyes that they could use to target specific proteins, specific biomolecules. They would stick to proteins so you can finally see microtubules. And they would, under UV lamps in the microscope, different wavelengths, you could see, as you see here on the left, the microtubules, the microfilaments, the intermediate filaments. You could see the individual meshworks. Then they started coming out with more and different dyes that would adhere to no longer just proteins, but now stick to different nucleotide structures, genetic structures, tightly packed, loosely packed DNA, and things like that. So you can now look at cells, no longer just staining them and try to figure out what's what. You can now go through and pick and choose. Stain it with this with blue, stain that with green, this with red, that with orange. And you take different pictures. Here's the blue, here's the red, here's the green, here's the orange, and you just stack the colors on top of each other, and then you get like what you see here. So you can now start to see everything going on in the cell. Now, if you think back, hopefully you remember cell and molecular biology, there was that endomembrane system. That in us eukaryotes, all internal membranes are linked. Not physically joined together, but are linked in that they feed back and forth into each other. The nuclear envelope is actually attached directly to the endoplasmic reticulum, the smooth ER, the rough ER. Parts of the smooth, parts of the rough will bleb off, become little vesicles, which will then traffic to the Golgi, become part of the Golgi now. Parts of the Golgi would break off and go to a nearest lysosome, become part of that. They just kind of fed back and forth into each other. And whenever you look at anything in the cell, you have to remember that there are levels of complexity. You think back to cell and molecular biology downstairs. Everything started with the monomer, nucleotide, an amino acid, a glucose or some form of a monosaccharide sugar. You started linking those together. You got a polymer, a macromolecule, DNA, some form of a polypeptide or a cellulose or something. You started wrapping those around each other and building bigger and bigger and bigger plasma membrane, enzyme, some form of protein enzyme complex. Here's they're showing, the different ways, the different things that come together, and those supramacromolecules come together to form the cell. So the levels of complexity. And all of these are held together, not always by carbon-carbon, bonds, but a lot of this is held together by some of the weakest bonds. Hydrogen bonds, ionic interaction, van der Waals, hydrophobic interactions. A lot of times what we're going to also be discussing, you'll see pictures of, you'll see discussions of, experiments of. And you have to keep in mind, in vitro, in vivo. In vitro is a lot of what we're going to be doing in lab for this course. Things done in glass, that's what in vitro means. Things done in a test tube, things done in a plastic well. It's artificial. It gives us an inkling of what's going on in the real world, yes, but it's still artificial. If it's done in the cell, in vivo, if that cell is still in the tissue, still in the organism, in vivo, in the living. So yeah, we'll do enzyme experiments, enzyme kinetics in the lab, isolated enzyme. The enzyme kinetics we find there are not always going to be the same enzyme kinetics you're going to find excuse me, out there in the real world, in the cell. Because in the cell, that enzyme is also competing with other enzymes for substrate. The cell is regulating that metabolic pathway by limiting substrate, by altering pH, by altering this, that, or the other.